Eran, I will start in a timely manner. So in a minute, when the clock strikes what I guess is 5 o'clock in Israel,
Yes.
then I'll begin and be grateful to you. I'll conclude my contribution. I'll introduce Phil Sharp and then listen as long as I can, but be grateful to you for hosting after that time the speakers to follow.
Will do, no problem. Thank you.
So it is time, and I'd like to begin by thanking all who are joining us. I'll mention what many of you would not know, that this is not a unique event, that we hold such an international school hosted, or I should say funded, at least in part by the Rothschild Foundation at the International Institute for Advanced Studies at the Hebrew University each year at around this time. These schools have dealt with many important topics in the past, including most recently last year, for example, G protein coupled receptors. The school was led in that case by, with contributions by Bob Lefkowitz and Brian Kobilka, and every previous school has attracted a great deal of interest and attention. The schools are mostly for the benefit of students, fellows, as well as faculty at the Hebrew University, but especially also students worldwide who, when it is possible to conduct the schools in person, are actually invited and often subsidized to come and join the meeting. Of course, this time it's being conducted in a virtual manner, and I owe a special debt of gratitude to Eran Mashor and Nissim Benevisti, not only for suggesting that we do this, but for making all of the arrangements that have made it possible. Thank you both very kindly. I will begin with a few introductory remarks to the topic, which has interested me for quite some time. I know that it's of a special interest to many people, especially as attested by the, I think, nearly 1,000 people who've registered on this particular occasion. I first became aware of the subject at one of these schools about, I'm guessing 5-6 years ago, when Dirk Gerlich spoke about the structure and function of the nuclear pore, and presented the remarkable finding that the counterintuitive capability of the nuclear pore, namely the remarkable filtration whereby large objects such as ribosomal subunits could pass through, but small objects like proteins on the size of, say, enzymes, bovine serum albumin could not, was explained by the presence of a hydrogel formed by disordered protein regions extending from the periphery of the pore. Dirk furthermore showed that specificity resided in the so-called FG repeats, and in particular, one imagines the interaction of tyrosine side chains, hydrophobic in nature, in an otherwise hydrophilic environment of the charged and hydrophilic disordered protein regions. Then Dirk performed what I think is one of the most beautiful experiments I myself have ever witnessed. He showed that by expression of a large quantity of those disordered regions, their concentration, and I believe it was actually done by precipitation in a dry state and then rehydration in an Eppendorf tube, the gel which formed, conical in nature, could be tapped out on the surface of, for example, a desktop or a piece of parafilm, and bathed in a solution of a fluorescently labeled protein, and the property of the nuclear pore replicated in that setting. What he showed was that a protein like a 75K serum albumin or what have you, or GFP or what have you, would not penetrate the gel, except if it included a nuclear localization signal and an important protein, which licenses the molecule for passage through the pore, in which case a wave of fluorescence could be seen to pass through that otherwise colorless gel. I made the mistake, even though I should have known better, of being astonished but not realizing that nature never wastes an important discovery of that nature. And I was reminded of my mistake not long after at another one of the schools, perhaps now 3 years later, if I recall correctly, when Steve McKnight came to report on the discovery of another example of the disordered protein region that assembled in a meaningful structure. In this case, the RNA transport granule formed by the Fus protein. These molecules consist of a positively charged region and a disordered one. The disordered regions assemble in a hydrogel-like particle with the positive charge on the surface. They can absorb RNA molecules and are responsible, I believe, for transport down microtubule pathways from the cell body to the synapse, so along the length of an axon. That work led to and Steve also discovered the basis of specificity in the collaboration with Rob Tikoff. Both Steve and Rob will speak at this meeting, so I won't take a lot of time to review what they found, except to emphasize the importance of their discovery of the basis for specificity. And I think it would be it would be worthwhile in introducing this meeting to call attention to what I view as important criteria for the significance of what are commonly termed condensates, clearly terribly important and components of cell biology in all aspects, an exciting new area which many have contributed to, and including especially those who've kindly agreed to speak at this meeting. As I say, the field has expanded rapidly, has attracted a lot of attention, plainly of very great significance. And I only want to emphasize at the outset two criteria I feel are of importance for, as it were, the validation of a finding along these lines and understanding at the molecular level, namely functional significance and specificity. Specificity clearly, because it would be impossible for every disordered region to interact with every other one, and obviously from even the only two examples I've cited, but many more that you'll hear about, as in every other aspect of nature and cell biology in particular, specificity is the watchword. With that, I will conclude this introduction and express a special gratitude to Professor Phil Sharp for his willingness to offer observations on this subject. I'm most anxious to hear his thoughts, as I'm sure everyone is. Phil, thank you very, very much for joining us. Greatly appreciated.
Thank you, Roger, and I'm honored to join. Let me share my screen, and then put it on full mode. Okay. I'm, as I said, honored to be invited and be included in this symposium or conference. I'm going to talk about condensate biology as viewed through the lens of transcription, but it is only one example of processes involved in gene expression that are almost certainly have condensates at their core. That includes splicing, it includes translational regulation in the cytoplasm, localization of mRNA, and then many cytoplasmic activities. The only conflict of interest possibly is I'm associated and advisors to companies that are involved in understanding phase transitions and how they might be, biology might be manipulated in their context. I want to cite this very early paper by Cliff Brandewine and Tony Hyman, who brought the dynamic properties, the liquid properties to our attention of these condensates. They were studying at the time germ granules and C. elegans, and they found that these were fusing and fissioning, and when you photo bleach them, they would recover in a half-life of seconds. And if you reduce the concentration of the proteins involved, they would become smaller in size, implying that they were in dynamic equilibrium, components in them with components in the cell. And similar observations about liquid phase transitions were noted about nucleolus. And now we know that there are a large number of membraneless bodies in the nucleus of the cell, as well as in the cytoplasm of the cell. I'll just mention nuclear speckles and perispeckles that are known to be associated with splicing and DNA damage, foci, stress granules, P bodies in the cytoplasm, emerging in increasingly strong evidence that synaptic activities and even membrane signaling might have properties of condensates. These condensates are formed by phase transitions, as Cliff and Tony informed us by the physics of those processes. These are all macro phase membraneless bodies or condensates. But if you take and fuse given genes, mediator involved in transcription, SR proteins involved in splicing, polyclone proteins involved in silencing, constitutive silencing out of HP1, fibrillin out of the nucleus, CTCF in forming TADs, what you see is that each of them form in live cells these condensates, which you can count and measure. And these, at this sensitivity, these are 50 to 100 molecules in one region of the nucleus, and even larger when you get to these larger contexts. And you can estimate that the fraction of the nucleus occupied by condensates is approximately 40%, 30% of the nuclear volume. Clearly, these are micronuclear condensates that can't be visualized by just staining cells with dyes. In a visit to Dallas, Mike Rosen pointed out some of the properties of condensates, and Steve McKnight as well. And I particularly like this experiment by Rosen and Parker, in which they mixed RNA binding proteins with intrinsic disorder domains and RNA, and showed that the association of these domains with RNA would form a condensate. You would picture that condensate being formed by a variety of interactions, let's say sort of millimolar affinity interactions between transient domains and the intrinsic disorder domain, micromolar or lower interactions out of RNA binding motifs, and they could be forming multiple valencies, and then RNA itself with even lower affinity interactions and being less dynamic. So many of the condensates in the previous slide also have RNA as one of their constituents. So we have to think of these condensates as mixed bodies that could have multiple components in them, and depending on their affinities being varied in dynamic properties. if we think about a simple model for condensates, and this model was developed with Arup Chakrabarty, and the intent here was to basically recast or cast the condensate process in parameters that biochemists measure. One is valency, how many points of interaction are there between different macromolecules? The other is affinity of those interactions, and then the number and concentration of a chain. And I'm going to talk about this model briefly just to point out something about the process. Here it's pictured as having multiple chains with sites that could possibly be modified, and at those modified sites you can get a crosslink. So you can vary the valency by the F parameter, the number of sites that are actually capable of being cross-linked, and the affinity of those sites in K on, K off. Now, if you limit this process to one cross-link per any two chains, this could be cross-linked to another chain, you can solve this analytically. And what that treatment suggests is that for a given number of chains, the larger, the lower the concentration, the forming of this condensate is inverse to the affinity of this interaction. As you would expect, higher affinity interactions, you would form condensates at lower concentration, but inverse to the square of the valency. Valency is clearly a major driver in this process. And you can picture this process as a condensate of chains going to a condensate that's associated with dynamic properties, and do a dynamic calculation of this transition dependent on these parameters. And Arup Chakrabarty and Krishna did that, and what's striking about it is its cooperativity. Small changes can stimulate a very dramatic formation of a condensate. Now, this is a condensate pictured theoretically for 50 chains. There's a twofold change in valency, and you go from 10% of the chains being in a condensate to 90% of the chain. And that's a Hill coefficient, which biochemists typically talk about, of about 6, a very large Hill coefficient. And if you're at a smaller number of chains, the Hill coefficient is lower. But even then, small changes in the properties and the concentration can make a very big difference in the formation of a condensate through these processes of a phase transition. Now, when I heard about phase transitions, and coming from talking to Rick Young at MIT, I was fascinated by his description of superenhancers. And let's talk about those, because I'm going to talk about the evidence that superenhancers are forming condensates. If you look at a typical gene, and this is in mouse embryonic stem cells, you see one place where you see OX, SOX, and NANOC. These are the transcription factors driving the properties of an embryonic stem cell. And this is a chip where you've immunoprecipitated out the protein and determined the sequences in which it's associated with. And you find that there's a site here with associated sequence transcription factors in the gene. And this is an enhancer, and it's associated with a given gene here, and it has polymerase and others at the promoter and at the enhancer. And we know these enhancers are typically transcribed in transient RNA. But here, Rick was describing clusters of enhancers, which he called super enhancers, and they were all driving the transcription of a given gene. And you see polymerase associated with them by CHIP, mediators associated with them by CHIP. You see acetylation proteins like P300. You see BRD4 that binds to acetylated histones, and deacetylation. So there's a number of factors all associated with these enhancers and the promoter, and clearly the enhancers had to be promoting the transcription of the gene. And what was really interesting about that is that these enhancers, all by three-dimensional ChIP-seq sort of assays, ligation assays, were associated with each other and with the promoter. And that if you compared them to a typical enhancer, where if you modulate this by mutation, you get a pretty linear effect, or you treat it by a drug that inhibits like BDR4. But if a super enhancer, you see a more cooperative activity out of both of those. And it's particularly interesting that these super enhancers are associated with genes with prominent roles in normal and disease processes, highly cell type specific transcription factors and oncogenic transcription factors as examples in cancer. So the proposal was that these super enhancers were forming a condensate, and it was quickly shown that you could see a condensate being associated with the gene in which is known to have a super enhancer. But I want to turn to the more dynamic aspect of this and turn to the work of Ibrahim Sissi, who's a professor at MIT. Here in live cell imaging of embryonic stem cells, he's fused mediator with GFP, and he's imaging that in live cells, and he's noting for there's a few stable clusters of mediator in these cells that you see fusion and fission, and he quantitates this over a period of time, and you can show that over about 5 minutes, these two condensates, a mediator, or Pol2 has similar effects, will fuse and then dissociate again over about 5 minutes, showing you the dynamic properties of these condensates in the nucleus of a cell. But I particularly like this experiment that he published earlier, in which he took a system that was well known, and that's quiescent fibroblasts, and you stimulate it by serum factors, and you induce transcription of an early response. And that transcription is known to peak at about 30 minutes. It's under Fos-June type control. And actin is one of those early genes. And he looked at this using live cell imaging, in which he labeled the gene actin by M13 insertion of RNA binding sequences. And that would tag RNA at the gene. And he labeled polymerase with a fluorescent fluor, and then used TC-POM to image, in which you then get high resolution and high spatial resolution. And you see here the actin gene labeled by binding to RNA. And you see here polymerase in a number of clusters. These clusters contain 40 to 80 types of polymerase. They have a half-life of about 5 to 10 seconds on average. Some are longer, some are shorter. And the interesting thing is that the actin gene co-localized with one of these clusters. And he then measured the half-life of the cluster and the number of mRNAs that are produced during the cluster formation associating with the gene. And what he found was a pretty linear correlation between cluster time and the number of RNA that were created and generated. He measured this by change in diffusion coefficient. And you see that the number of nascent RNAs peak about 15 minutes after stimulation, as we have known from biochemistry over a number of decades, and the cluster time peaks. So in a quiescent cell, it's on 8 seconds in the cluster, and when stimulated to produce a larger number of polymerases, it goes up to 25. Again, suggesting this dynamic relationship between the burst of transcription and the activity of association of a condensate with a gene. And you could easily picture the condensate concentrating polymerase and other components to give you an initiation. So we talked about super enhancers and valencies. how sensitive, and then the burst of transcription being associated with the formation of a condensate associated with the gene. These proteins that are involved in condensate formation typically have intrinsic disorder domains. This is shown here for mediator here in BRD4, and about 30% of all genes have intrinsic disorder domains. And before this idea or concept of condensates, we had very little insight as to what these domains meant to biology. Here's a mediator, intrinsic disorder domain. These are the amino acid sequences in the protein, and you see this cluster of serine. You see serine being clustered in the mediator is conserved back to amphibians. And if you take the mediator and intrinsic disorder domain and fuse it to GFP, you put it in a concentrating Fical solution to concentrate the protein, you see condensate formation. If you mutate the serines to alanines, you don't. That doesn't say that the specificity is coming out of serine. It's basically part of the properties of this protein that form the intrinsic disorder domain. But what is interesting is if you think about a super enhancer, you should picture it in this fashion with a series of enhancers that bind transcription factors. And you see a cluster of them pictured as forming a condensate. The transcription factors have a DNA binding domain of high affinity interacting with DNA and an intrinsic disorder domain. Mediator has an intrinsic disorder domain and activity structured domains, and they interact. That intrinsic disorder domain will commingle and co-form condensates with the transcription factor intrinsic disorder domain, and the carboxy terminal polymerase, a polymerase, does as well. So what you picture here is a condensate forming, specified by the binding of transcription factors to DNA and clustering at a valency. These intrinsic disorder domains associating with one another, forming a condensate at a critical concentration and valency, and then concentrating these factors to direct a burst of transcription from the polymerase. And then after the polymerase burst of transcription, this dissociates in most cases, and then under non-equilibrium properties will reform in different ways. And others will possibly talk about what the nature of that process is. But if you think about this as being a stable entity that stabilizes these interactions, you can picture it in terms of valency. And if you take a piece of DNA with low valency, let's picture it's where separated binding sites for a transcription factor, or higher valency, typical of the experiment with RNA, you mix DNA OC4 as a DNA binding factor with its intrinsic disorder domain, mediator with its intrinsic disorder domain, and the low valency DNA will not form under these conditions a condensate, but the high valency does. And if you take the same type of valency of transcription binding sites and fuse it downstream or upstream of OC4 promoter driving luciferase, you can see that with multi-binding sites, you can get a dramatic stimulation, almost step function from 4 to 5 in this transient order domain. So we've talked about transcription factors forming a condensate, driving a burst of transcription, this condensate having dynamic properties of fusion and fission, didn't show you photo bleaching, giving rise to initiation of transcription. We know upon initiation of transcription, the carboxy terminal tail of polymerase is phosphorylated. We know that if that is phosphorylated, it'll no longer mix with the condensate formed by mediator and others, but it will form with condensates formed by serine arginine repeat proteins. And we've known for decades that these proteins are concentrated in speckles. with the dynamic properties of condensate type activities. There's a large host of these proteins, some that bind to RNA, some that do not, and they contribute to the splicing. So we picture a condensate forming not only of transcription, but during the splicing process, giving rise to organization of the spliceosomes, and after splicing to the transport from the nucleus to the cytoplasm through the nuclear pore, as Roger introduced us to condensate properties of the FG proteins in nuclear pores to give rise to RNA that then mediates, is regulated in the cytoplasm by condensate type processes. So I thank you for your attention. This work has been done in collaboration with Rick Young and colleagues who are mentioned here, Arup Chakrabarty, physicist in his graduate student Krishna at MIT, and Ibrahim Sissi, who's published independently and whose work I stressed in the terms of the dynamic and visualization of the properties of condensates. I thank my colleagues over the years who's contributed to my research and some to this research, and I thank you for the opportunity to give this talk. Thank you.
Thank you, Phil, for this wonderful presentation. I think the stage is set for the meeting. I will turn the leadership of the meeting now over to Aaron Mashore.
Thank you, Roger, and thank you very much, Phil, for this grand opening. So we're going to have a discussion at the end of tomorrow. So for the sake of time, I think please keep your questions for the discussion. And I just remind the speakers that we have sessions that are 20 minutes in length, and we are trying to have 3 minutes for questions. If we won't have this time just because of the time differences and the fact that in some places in the world it's very late at night, we're really trying to finish on time. So if there is time for a question or two, we'll take them. If not, please keep them for the discussion session at the end of the meeting. So I'm very happy to welcome Amy Gladfelter from the University of North Carolina, Chapel Hill, and she'll be telling us about RNA sequence specificity in condensate assembly, relating to what Roger just told us about function and specificity. So thank you, Amy. Please.
All right, super. Let me just get myself presenting here. Can you see my screen?
Yes.
Okay, super. Great. It is such an honor to be here to share our work thinking about RNA-driven phase separation. And today I hope to talk about work both in cells and also relevant to the SARS epidemic. So my interest in condensates emerged from a deep interest in syncytial cell biology. So syncytia are ubiquitous multinucleated cells, and you can see an example of a filamentous fungus growing here under the microscope, where each of these white spots is an individual nucleus sharing a common cytoplasm. And if you went out into the forest and looked into this under the soil, you would see networks of cells that look like this. But of course, they're also found throughout our body and throughout the biosphere. And they present some really fascinating cell biological, cell organization problems that have really driven a lot of my research over the years. And they've led us to think a lot about the fundamental structure and organization of the cytoplasm, which we know is a densely packed and dynamic environment. And the question really is, at the frontier of cell biology, how does order emerge within this complex milieu of molecules? And the principles of phase separation have really guided us into a whole new way of thinking about organization at this level. And in particular, in our lab, we've been able to appreciate that condensates create particular patterning or neighborhoods within these very large networks of multinucleated cells. And that essentially by spatially localizing RNAs and proteins, this enables these very large cells to be highly multifunctional, despite the fact that there is a large continuous cytosol. And this has been a very powerful system for understanding how cells control where and when condensates form, and also what are the determinants of molecular specificity or composition, which Phil already touched upon this really critical problem, because one would predict based on the physical properties of condensates that they should essentially fuse together and create one gigantic mixture. However, we know that in fact there is a tremendous number and variety of different condensates that coexist within the same cell compartment. So we've been able to make some insights into how this works by studying cytoplasmic RNA-based condensates that control translation in the cytoplasm of filamentous fungi. One of the key proteins we focused on is an RNA binding protein that has a low complexity sequence that's composed of a polyglutamine-rich sequence. It also contains RNA binding capacity through folded RRM domains. Now, what we've been able to see is that this protein assembles into multiple distinct kinds of condensates in the cell. In particular, they're important at sites of polarity, both for initiating new sites of polarity and maintaining sites of polarity, but also for patterning within the inside of the hyphae here for determining when and where the nuclear cycle happens. And so they're controlling also the cell division cycle. Now, this protein is very discerning in terms of how it demixes in vitro, in that under physiological conditions, it doesn't form condensates on its own. It absolutely requires very specific RNA sequences, and it only demixes with its native target sequences. And most notably in this system is that if you can visualize in this column here, if condensates are made out of different targets, so in this case, a condensate is made by mixing this protein with an RNA important for cell polarity, or in this case, we've added RNA important for the cell cycle, you can see that these form actually two distinct populations of immiscible condensates. So this really suggests that there's features in the RNA that is making phase separation both sequence specific and is a molecular basis for this immiscibility. So we've been addressing mechanistically how this works, but I really want to emphasize thinking about RNA and all the properties that it brings to condensates. So in addition to having a very uniform negative charge in contrast to the protein components, it also is a source of multivalency in the system with sequence encoded binding sites. RNAs are incredibly large compared to polypeptides and can be highly variable. And what I hope you take from this talk today is the importance of assembling dynamic structures through both intra and intermolecular base pairing. And I think a very unappreciated feature of the RNA is the mechanical properties that it brings, and the fact that RNA, just like peptides, has both disordered and ordered, highly structured elements, and that the interplay between structure and disorder is also highly relevant, we think, for RNA components in condensates. So we've been able to see that RNA structure is critical for specifying interactions that determine both the composition and the material properties of condensates. So one way that we've been able to see this is taking advantage of mutational profiling technologies, where we're actually able to empirically model RNA structures and also protein interaction sites. And so quite simply, this approach allows us to reveal what areas of an RNA are likely in single-stranded versus in base pairing interactions by a small molecule that will modify these bases that are exposed, that can be detected through deep sequencing, and then essentially lead us to predicted structures. So using this approach, we were able to identify a mechanism grounded in RNA structure for explaining this immiscibility phenotype. And specifically, what we could see is that these two different kinds of condensates, those for the nuclear cycle and those for cell polarity, do not mix with each other because of sequence-encoded RNA-RNA interactions that ensure that cell cycle RNAs co-condense and that polarity RNAs co-condense. And we're really actively now still trying to understand these kinds of contributions of RNA at the levels of both molecular and physical identity. And I really want to emphasize to the audience here today that we have a huge opportunity here as a field to understand a major source of information that I think is relevant to molecular identity. And I encourage everyone to be thinking about analyzing specific RNAs in their experiments. Now, I want to take the second part of this talk here to really focus on a new project in the lab, but that also is grounded in this idea that RNA sequence is critical for specificity and phase separation. And it's a question that we started asking really at the onset of the pandemic as our labs were shutting down. And this is a question related to the SARS-CoV-2 coronavirus, really focusing on understanding how the nucleocapsid protein can specifically find, bind, and package the genome. And our fascination in this was twofold. One is if we consider the virion, we know that the N protein is coding the genome, and the N protein has features of a condensate driving protein in that it has a high degree of intrinsically disordered sequences, but it also has the capacity to bind to RNA. And of course, there's this magnificently large RNA-based genome that really captured our imagination in thinking about this as a phase separating system. And indeed, if you mix together the N protein with genomic RNA, you can see readily this concentration dependent phase separation occurring. So indeed, we could also see that the N protein was able to form condensates within the cytoplasm of mammalian cells. And you can see here in this movie that they have liquid-like and fusing, liquid-like properties and the capacity to fuse. So we've been interested in understanding what features of the genome drive this condensation. And really, the big question that's been fascinating us is how does the virus package one copy of its genome and exclude other non-specific RNAs that are obviously abundant in the cell? And finally, we've also been considering the temperature dependency and the role of RNA in tuning the temperature at which this protein phase separates. So we were interested to know if phase separation was being driven by specific sequences in the genome. And specifically, we focused on essentially 4 different regions of the genome. These are areas that were either under synonymous substitution rate constraint or had been linked to coronavirus biology in SARS-1. And so the results of our initial experiments here were so surprising to us, but what we could find is that when we synthesize RNA encoding, or rather from the 5' end of the genome, the frame shifting region, an area that is shown to be sufficient in SARS-1 for packaging, or an area at the 3' end, which encodes the nucleocapsid itself, we could see very different phase separation behavior with the nucleocapsid protein. So specifically, these RNA sequences at the ends of the genome were driving phase separation. And you can see that here with these condensates formed in this column and in this column here. In contrast, RNAs from the interior of the genome actually limited phase separation and actually seemed to be counterproductive to any phase separation. So we have opposing behavior in the genome from different RNA sequences. And I want to emphasize that in particular, for example, the 5' end and the frame shifting element, these RNAs are the exact same length. So this tells us that there are differences at the RNA sequence level that are promoting different phase separation behavior. So we wondered what these were. So we again turned to a mutational mapping approach. In this case, we adapted a technique with Kevin Weeks and Mark Bernanke at UNC called RNP map, which allows us to detect crosslinks of the protein to the RNA, along with determining the structure of the RNA. And what we could see when we looked at a sequence, the 5' end, that promotes phase separation, is we could see these very discrete binding sites. These were the strongest binding sites that we saw for the N protein binding to this sequence. And if you looked across the entire sequence here that drives phase separation or promotes phase separation, we could see these very discrete binding sites for the protein. And we're right now trying to understand what the element is they're recognizing, but we think it has to do with these very strong structures, these helices adjacent to this site. In contrast, when we look at the frame shifting element region, first you can see that this is a highly structured sequence compared to the 5' end, but here we can see the N protein is binding nearly uniformly along the sequence, as if it's wetting it or coating the entire RNA sequence. So we see a very different binding pattern depending on these different RNAs. Here we can imagine these almost specifying very specific valence, where in this case we can almost imagine the RNA is in essence solubilizing the N protein. We wondered about the other features of the genome with Rachel Silfon at Princeton. We just compared all the sequences in the genome to these areas that are predicted to drive phase separation, and could see that indeed most of the features, the sequence features, seem to be contained within the 5' and the 3' end, whereas many of this interior area sequences appeared to behave very similar at the sequence level as the frame shifting region. And indeed, when we did RNP map on this area that limits phase separation, again, we see this uniform coding, very similar to what we saw with the frame shifting region, whereas with the nucleocapsid sequence that promotes phase separation, we again see these very discrete binding sites for the N protein, very similar to what we saw at the 5' end. And so this leads us to a model where we think that there's actually multiple different ways the N protein is interacting with the genomic RNA. In particular, we think there are very specific encoded binding sites within the 5' end and the 3' end that are providing particular valence that is driving the phase separation process. In contrast, we think there are many sequences within this interior part of the genome that we think may be providing a more sort of non-specific or electrostatic effect on the phase separation, essentially lubricating the condensates. And we think this may be very important given the enormous size of this genome to prevent gelation of the RNA, at least until potentially gelation could occur at a later time point. But we imagine at some point in the process that achieving some sort of liquid-like state is important, and that these interior sequences might be relevant. We speculated that having this sort of optimum combination of sequences may be important in packaging, and that this might be a way to help exclude non-specific RNAs, but include specific RNAs, particularly the genome. So we have done some experiments that address this, and I'll just talk about one of these here, where we asked essentially whether the material properties of a condensate might be important for promoting specificity in packaging. So we made droplets out of the nucleocapsid protein with genomic fragments. So these are fragments that aren't translated. And we asked whether sequences that were genome specific could enter into these condensates, or whether sequences that are subgenomic, so these are RNAs that would not normally thought to be packaged, if we mix these together, would they be able to enter? So we wondered whether certain sequences would be included and others excluded. So here's an example of one of these experiments. These are these N protein droplets made out of genomic fragments, and we, in the top, added genome-specific RNA. So this is RNA that's like what would be packaged. And on the bottom, we added subgenomic RNA, and this is RNA that we would expect would be excluded in a packaging context. And interestingly, when we did this experiment, we could see that RNA that was genome specific actually mixed readily into these condensates, whereas subgenomic RNA actually tended to be excluded from the condensates and actually would form its own distinct condensates. And I don't have time to show this, but I can say that in fact these different RNAs produce condensates which have very different material properties based on fusion and on FRAP. So we think one model for using condensates within the viral life cycle could be to create compartments that will be specific for including or excluding specific RNAs. And I'll just close in the last minute or two here with a second packaging challenge, which I think is so fascinating, which is how is a single genome put into a capsid? And experimentally, this is a tough, it's tough to prove experimentally. So right now we're just looking at this using molecular dynamics simulation. And what we're doing is we're applying a coarse grain simulation where we're representing the N protein here as just two beads that have the ability to interact with themselves, but also to interact with the RNA genome, which we're representing here as these beads you can see here. And really the question is there something about the spatial patterning of the specific interaction sites at the ends as opposed to in the middle? And can we begin to understand what would drive singularity in packaging? And so we're taking advantage of Lossy, which is a Monte Carlo simulation engine developed by Rohit Papu's lab at WashU. And importantly, this allows for either very specific valence interactions or multivalent interactions. So this is just some initial results from this, these simulations. Importantly, I should also say in Lossy, you can actually create phase diagrams. And so you can know when the system is actually undergoing phase separation. And so you can actually get a phase boundary here from the system. So what we could see when we started mixing together proteins and RNAs is initially, depending on the stoichiometry, we would just end up with all the RNA molecules clumped together into one gigantic assembly. And I should say in this heat map here, we're looking at the number of RNA clusters where these light colors are where the cluster number is getting higher, which is what you would want to achieve in terms of separating the genomes and packaging. Now, what we did is we started increasing the number of proteins relative to the numbers of RNAs. And what was really exciting is as we increased this ratio, what we could see is that eventually we did achieve separation. And you can see here separation of the RNAs. The RNAs are now each in their own individual clusters. And what's exciting about this is that this actual ratio is equivalent to the numbers of copies of N protein that are thought to be present per genome in a virion. So this is all theoretical, but our current work is trying to think about ways to understand how the condensation process may be helping to drive singularity in packaging. Now, in this last second, I just want to say we have a lot of interest also in understanding how phase separation may be relevant to the infection cycle of COVID. And we started thinking about a role for temperature in this. Obviously, temperature can be a modulator of phase separation. And there's very interesting just biology here in the vector of bats, which have to lower their body temperature in order to hibernate and raise their body temperature when they go out and spread viruses. And there's also temperature gradients existing in the upper airways of humans. So this prompted us to examine the role of temperature in phase separation of the nucleocapsid protein. So I just show you a bit of data here on this. So the N protein on its own actually will undergo phase separation at high temperatures. So it has what is called a lower critical saturation temperature. But what I think is really fascinating is that the presence of RNA can actually lower the critical temperature here. And depending on which RNA is included, this actually changes the lower critical temperature. So both RNA and protein, we think, are contributing to the temperature sensitivity, and we're very interested to know what RNA sequences are relevant for promoting the temperature sensitivity. So I'll just wrap up now with this final model that we think that features in the RNA genome are really critical for determining both the phase separation behavior and the material properties of condensates. And this process is temperature sensitive, and we think that this could be a point of intervention with drugs. I want to highlight there's a lot of really exciting work on the bioRxiv on SARS-CoV-2 phase separation. I'm just listing some of the other papers that have been posted over the last few months. And I hope that after this talk, everyone is thinking a little bit more about the role of specific RNA sequence and structure in really understanding how mesoscale physical properties arise from sort of these nanometer scale interactions, and that RNA is a rich source of information in condensates. So I want to thank my team who did the SARS work and FAST grants, also for supporting the SARS work, and also current people in my lab who contributed today to the story, as well as funding agencies that support all of our work. Thank you for your attention.
Thank you very much, Amy, for this fantastic talk. And again, for the sake of time, we're already a few minutes behind schedule. We'll go on. So again, please save your questions to the discussion at the end of the meeting. And also you can send questions to the chair of each session for discussion at the end of the talk. It will be easier maybe to handle. So with no further ado, we'll continue with Eilon Sherman from the Hebrew University. And Eilon will tell us about nanoscale protein complexes that regulate cell T-cell signaling. Please, Eilon.
So thank you, Eran, and the organizers. Let me share my screen. Okay. So hopefully you can all see it. And indeed, I'm going to talk about how proteins come together to form complexes. And these complexes are at the nanoscale, and they can regulate cell signaling. And the major focus of my talk is going to be on immune T cells, but I'm going to try to make it a little bit more general. So if you consider cell signaling, you can see many different arrows coming together and linking these different proteins, effectors, receptors, etc. And obviously, this model is a very huge simplification of what is going on at the molecular level within a cell, since all of the interactions are stochastic. And also, this is kind of a well-mixing model where the concentration of molecules is equal all over the cell surface or the cell cytosol. And obviously, this is not true. So on the other hand, we have this view, and Phillip has shown this slide before. So I'm going to focus on these protein structures, such as membrane clusters and signaling puncti. And also, I will touch on synaptic densities. And I'll try to show that these are quite ubiquitous. So typically what we do in our lab, we take the cells, and these are, for instance, T cells, we express fluorescent proteins within, and we drop the cells on surfaces such as these. And these surfaces might activate the T cells or not. And now we can visualize the interface between the cells and the cover slip. And this is a confocal image of such an interface. This is a single cell. And you see here with 3 different fluorescent colors, 3 different proteins. For now, the nature of the proteins, the identity is not critical, but you will see that this molecule is kind of a hub for many different proteins, many different effectors that come together or bind this protein upon cell activation. And what you need to take from this image is that it's very colorful. So it's very heterogeneous. And each and every point here is a manifestation of many different complexes. In fact, each pixel is about 200 nanometers in size. So it can accommodate millions of different complexes here, all have different stoichiometries and their function. So ultimately, we would like to be able to characterize these heterogeneous structures at a single molecule level. And for that, we turn to super resolution microscopy. And in this case, this is called single molecule localization microscopy or photoactivated localization microscopy, which is a variant of that. I will not have time to talk about how we do this imaging. All I will say is that Eric Betzig, the inventor of this, got the Nobel Prize in 2014, and that this method allows you to visualize individual proteins, such as this molecule, the surface molecule that I showed before. And this is again the footprint of a cell touching a cover slip. But now you can zoom onto any of these clusters that you see here, and you can start counting molecules. And each and every molecule is localized with a resolution down to about 20 nanometers. Okay. Now, our purpose here is to characterize first the structure of these condensates or protein aggregates. And for that, we turn to statistics. And for instance, we can measure the clustering of these. Now it's a point pattern of molecules. And the bold line here is the data. However, this should be contrasted with a null hypothesis of randomness. So these two fainter lines are due to a model where you take molecules and you throw them on a wall and they stick randomly. So this would be called a Poissonian process. And you can see, and these are the 95% confidence intervals, and you can see significant clustering, right? Okay, that you could see from the image, but now you can actually quantify the typical sizes of the clusters, and you find that they are much smaller than the diffraction limit of light at 200 nanometers. So these are really tiny structures at the nanoscale. We can also employ a clustering algorithm, right? You can identify these clusters, and now you can build a distribution of sizes now in copy numbers, right? And this is a cumulative distribution. So you can appreciate that about 70% of all clusters are actually dimers. And then another 10% are trimers, and so on and so forth. And only a very small fraction is manifesting the very big, more visual complexes, right, with dozens of molecules within. Okay, so these typically have continuous distribution of sizes, and they go down to dimers and trimers, and they are at the nanoscale, so we call them nanoclusters. Now, we are very interested in signaling in protein-protein interactions, right? This is what conveys the signal. So we took this protein and a very strong effector protein that binds it upon activation and phosphorylation. And again, you can see the clusters, but now you can see that with two colors, we can identify this kind of homogeneous mixing of the two proteins within the complexes. And this can be captured now with what is called bivariate pair correlation functions. This is the bold line. And the null hypothesis now is that indeed these two proteins mix homogeneously within the clusters, right? So we can now capture strong interaction between a protein and an effector. Now, if the two proteins are not related, such as in the lower panel, then this correlation is flat. And this is indicative of having no correlation. And you could also have effector proteins that have partial interaction. So it's not null, but it's not homogeneous, right? So now we can also quantify the strengths of the interaction. Now, adding a third color, now we can monitor higher level of interactions within these condensates or complexes. And now you can see, say, two effectors in blue and red binding the green cluster. And you can see them even cluster partially within the clusters. But now we can get to ask questions like, are these interactions cooperative? And I do not mean cooperativity by the biochemical sense, but by an analogy or a broader definition of cooperativity. For instance, if you consider the red protein, you may ask whether its binding to green is assisted by the presence of blue proteins, right? And we came up with these fancy statistics, never mind the name, but basically it captures this excess cooperativity if you have the presence of blue next to red and green. And indeed, you see that red is assisted by blue to get to green, and also symmetrically blue is assisted by red to get to green, which is nice, right? Now, is this always the case? Is this always symmetric? And the answer is no. So basically, if you consider other two proteins, such as actin here, which might nucleate these complexes, and this binder, this effector SLIP76 in red, you see that red indeed is promoted by blue to get to green, but blue doesn't care about the presence of red to get to green, right? So this is non-symmetric, and this can only happen in an environment that is not in equilibrium, right? And it suggests a sequential deposition of proteins within the complexes. And I will not go into too many details. This is now published. So overall, we get a sense of what is going on upon cell activation. And I will talk in a little bit about this receptor clustering, and then these nucleate into complexes that overlap. And now this, this protein, adapter protein that I have been talking about, nucleates these larger complexes and binds many, many different effectors. And people have counted about 24 of different effector proteins. However, surprisingly, these coalesce into these structured complexes that are decorated by actin. And now you can contrast this to these cartoons that people have in mind typically from biochemical assays, for instance, that are simplifying this much richer image. So now I get to tell you a little bit about the cell system here, which is T cells. And T cells are mediators of the adaptive immune system. And they face the task of searching for cognate antigens on the surface of antigen-presenting cells. And typically they would look for foreign pathogens, right? Fragments of the pathogens, and they need to distinguish them from self-antigens, right? Otherwise we get sick with autoimmune disease. And this decision-making needs to be very balanced. It's highly sensitive on the single molecule level. It's highly specific, as we all know, and it happens within seconds. So it's hugely efficient on an overwhelming background of non-specific self-peptides. And this is still an open question in the field of how information is being processed to give rise to T cell activation. And some say that in resting cells, you have this conformation of the T cell receptor that needs to be changed upon ligand binding, as you can see here. Also, you start having this clustering, maybe, and some people say even that you can actually push out from the tight contact glycoproteins that are fairly bulky and also act as phosphatases that tend to quench the signal of the T cell receptor. Okay, and even you might have a bystander effect of cooperative activation of neighboring receptors. Okay, so let's see if we can capture some of these processes, and mind you, these processes might be very dynamic. Okay, so let's start with clustering, and indeed we turn to live cell imaging now with super resolution, and again, each point here is a single molecule. In green, receptors. In red, these are effector proteins that only get to the membrane and bind the TCR, T cell receptor, upon cell activation. And you should know that the activation field here was very homogeneous. Okay, we made sure that it's 10 times more homogeneous than the smallest picture that you can see. And this is important because now each and every green point, any receptor, has the same probability of recruiting a red dot, an effector, right? Only what you see is that at some places, at some times, a bunch of receptors fire together, okay? So you have a localized activation of T cell receptors that happens at specific times, specific places, specific times. And this is a very strong manifestation of cooperativity and even long-range information flow between not just neighboring receptors, but also receptors that are fairly far apart, about 100 nanometers. So we see a lot of cooperativity within these clusters, and surely we could quantify this excess of clustering, this activated activity or clustered activity of TCRs and its dynamics over time. And I will not go into too many details. But I do want to look at another mechanism, and this segregation of the glycoproteins is called kinetic segregation. And let's see if we can capture that. So this is a T cell that falls on an activating cover slip. And again, receptor is in green and the glycoproteins are in red. And what you can see, each point is a single molecule. And soon enough, you see that red proteins are pushed away from the receptors that are fairly clustered. And I want to focus on just a single contact here, like so. And all of this is a few hundreds of nanometers in size. And you can even notice this black region, this black gap, which is due to the limited membrane flexibility. This is just mechanics. And if this is just mechanics, maybe we can understand it using a mechanical or a statistical mechanics model. So we embed these proteins in two grids, we let them diffuse and interact, and we can assign the potentials of the interactions between the proteins of the two membranes. Some of them are glycoproteins, these integrins, the receptors and ligands, and so on. And with a model that is now published, and you can access it through this link, you can start capturing both the membrane height, here gray shaded, and also this patterning of the molecules and the kinetic segregation. And also we can do this trick. We can take information from the experiment and plug it into the simulation and make a hybrid and see if we can actually predict what is going to be the localization of some molecules. For instance, here we took the green positions and we try to predict the red positions of the molecules, the glycoproteins, and you can see that we are doing a very nice job in capturing the patterns that evolve over time. So I told you a little bit about signaling complexes and how they relate to these very unexpected structures that we could find at the membrane. Also, I talked to you about the patterning of these molecules, which is highly dynamic and absolutely has a functional role in TCR signaling. But now I want to make it a little bit more general. So we also started looking at other systems. And for instance, here, these are excitatory cells where you need to release material, say hormones or neurotransmitters, but this should be done very fast upon vesicle fusion. So there was this hypothesis of our collaborator, Daphne Atlas, that maybe these calcium channels here can talk directly to these proteins that decorate the vesicles. These are called syntaxin 1A. And indeed, you can see them on the left, nicely interacting at the membrane. Okay, so yes, you do have some strong interactions of these two. Now, what about viruses? As with the previous talk, we are also interested in that. And viral assembly is also a type of a phase transition. So for instance, GP41 is a code protein of HIV-1, and it needs to assemble at the membrane of a T cell before the virus can bud out. So it can bud and be released. And what we unexpectedly found is that this code protein, GP41, likes to associate with the T cell receptor. And it also influences its signaling. So this is again published, and I will not spend too much about that, but I do want to show you some very recent data from the lab. And this is about aberrant Ras signaling in melanoma. So in melanoma, we all know that you have this EGFR, receptor tyrosine kinase signaling that activates the mitogenic pathway. and these go through RAS and RAF, MEG, ERK, etc. Now, RAS gets highly mutated in about 40% of the melanoma cases, and this incurs constant activity of RAS and activation of RAF. And it's undruggable because its affinity to its ligand is way too high. So we started imaging this RAS in melanoma. It's NRAS and RAF proteins and interactions. And again, time and time again, we find proteins in clusters, right? So these are complexes, very dense, and you can see them not associating without cell activation, but surely upon EGF activation of the cells, you have abandoned NRAS recruitment to the membrane. And now, mind you, BRAF is still segregated or still clustered, but now red proteins, NRAS, can associate with these clusters of proteins. Now, we also try to look at mutated NRAS that confers melanoma, very aggressive melanoma, and you see that without cell activation, you have many clusters, and what is really amazing is that the cells are finding ways to get NRAS and BRAF associated while they are still highly clustered, okay? So this is a very different phenotype from EGF activated cells, but still the cells know how to convey the signal. Okay, so with this, I would like to conclude. And again, I showed you a little bit of the structure and cooperativity of these very rich condensates. And these are highly dynamic and clusters of proteins that are formed by stochastic processes. I could show you how phase transition and segregation of proteins could help TCR signaling. And finally, I was trying to make it a little bit more general. And actually, we see time and time again proteins co-localize in clusters, in mutual clusters, in a way that can convey and regulate signals. So thank you for your attention.
Thank you very much, Eilon. So again, for the sake of time, we're every time running another 1 or 2 minutes behind. We'll continue. And again, save your questions for tomorrow's discussion session. So our next speaker is Tony Hyman from Max Planck Institute, Dresden. And he'll be telling us about pre-wetting transitions, information of condensates on DNA.
Sorry, forgot to turn on my So good afternoon, everyone. It's actually a great pleasure to be able to talk in this meeting, especially since this year I just took up my Israeli citizenship. So this is the first talk I give, at least virtually, in Israel, actually as an Israeli citizen. And as we have heard already in this meeting, you know, the many compartments in cells are described as biomolecular condensates, and many that you can see here are sort of individual condensates inside cells. But what a picture that also shows is that many of these condensates are actually associated with surfaces, such as the membrane or the nuclear envelope. And in fact, the cell is full of surfaces. It'll have polymers, such as microtubules or actin. It'll have membranes. And all of these cases, there's a rich and interesting biology about the way that condensates interact with surfaces. And so if we think about these liquid-like condensates, it's very interesting to think about how they interact with these surfaces and how that affects their biology. And what's particularly interesting is that these surfaces are different. A microtubule is a stiff rod. A membrane itself is fluid, and so it's a liquid-like condensate interacting with a lipid membrane. And over the years, we've sort of bumped into these surface condensate interactions. For instance, in our first paper on condensates and P granules, we actually used the fact that P granules wet onto the nuclear pore as a criterion for saying that we thought they were liquid-like, a little bit like, got here, glycerol wetting onto a coated bead. when we worked on microtubules, here's an experiment, for instance, where you take a drop of the microtubule associated protein tau and you add tubulin and GTP, it'll partition into the drop. And when it does so, it will then form microtubule bundles inside the drop. And you can see the way this interaction works particularly nicely if you add some heparin to de-wet. You can see the drop then de-wetting from the microtubule bundle. And so the way that condensates interact with these surfaces is like to be a very interesting source of physics. And indeed, it's very well studied in soft matter physics, but not that well studied actually in biology. And so it's unclear how we can adapt the soft matter physics ideas to biology, because generally they tend to work on larger surfaces with more molecules, where here we've got smaller polymers that are more complicated and interacting with complex proteins. And we decided, based on the pioneering work we've heard about from Rick Young from many groups at MIT, to try and think about the DNA as a surface and think how the surface interaction can affect its properties. Now, by us, I mean 3 talented postdocs and from the labs of Stefan Grill and Frank Ulica. And that's Sina Whitman, Sandeep Chaudhary, and Jose Marin. And the question they wanted to think about was, how is it that condensates interact with DNA? And for this purpose, they picked a pioneer factor called KLF4, cripple-like factor 4, which is one of the Yamanaka factors involved in cell programming. It's involved in development and cell identity. And here are some references referring to the work on discovering KLF4 and its activity. Now, you can make KLF4 in a test tube. Here's a nice gel of purified KLF4 GFP. And like many of these proteins, when you then put them into a test tube under the right conditions, you then make liquid-like drops. And here we're seeing liquid-like drops forming from KLF4. They have all the properties of liquid-like drops. You can see fusion on the right. You can see photobleaching recovery on the left. And there are many other properties that show they form classic liquid-like compartments. Now, for any protein that actually phase separates, you can also measure a saturation concentration. In this case, they measure saturation concentration of 1.2 micromolar. So what does it actually mean to have a saturation concentration of 1.2 micromolar? Well, if you look at a typical phase diagram, it's what's called a binodal. LLPS means liquid-liquid phase separation happens in the dark green area. And you can define a saturation concentration at a certain temperature. In this case, it would be 1.2 micromolar. Now, but what is the concentration of these proteins inside a cell? If you measure the in vivo concentration of KLF4, these are kind of tricky measurements, but it's definitely lower than 1.2 micromolar, maybe 0.5 micromolar. Many of these transcriptional effects are actually quite a lot lower than these saturation concentrations you measure in vitro. So, but if you add DNA, you can stimulate condensate formation at much lower concentrations. And Phil talked about a nice experiment from Tribulus using DNA-sequence, which is a sort of more sophisticated version of that. So we know that at low concentrations, you can stimulate condensate formation. And so the question is, how actually is the DNA surface helping to form these KLF4 condensates? So to study this, they set up an optical tweezer assay to look at the problem of condensation on DNA directly. And this consisted of taking a piece of lambda DNA that's 30 kb long, holding it between 2 traps. And there's a whole set of microfluidics that can be used to change buffers. So you've got a bit of DNA, you can look directly at condensation, and you can change proteins, you can change buffers, and directly observe the processes. And when they did that, they saw beautiful formation of condensates on DNA. And that's shown in this picture. We've got about 100 nanomolar KLF4, and you can see here that you're forming little condensates on the DNA chain. And if you do some quantitation, you come up with a minimum of about 1,000 molecules of KLF4 in each one of these condensates. It's probably an underestimate, and it may be a lot more based on fluorescent quenching. So you can also study the dynamics of the formation. So what you're seeing here is on the y-axis time, and on the x-axis you're seeing position on the DNA. And you can see after you flush in KLF4 with microfluidics, you can now begin to see the condensates forming on the DNA. And on the right-hand side, you're seeing that these condensates are fused, as you might expect, so they're quite dynamic. Here you're actually seeing a sort of close-up of some of the formation of these condensates on DNA. And they've done a lot more experiments using tools such as FRAP to really demonstrate that these are very dynamic condensates on DNA. So what does this mean? Well, let's go back and think about our binodal, which is that what's now happening is that this black dot illustrates that condensates are forming outside the classic binodal where you expect phase separation, and it's surface induced. And so that means that somehow the DNA is influencing the phase separation away from the classic binodal that you expect. So it turns out that in soft matter physics, there's a concept called prewetting, which is a surface-based effect, which extends the binodal to the left into the areas you wouldn't normally expect condensation. And this defines sort of two different bound forms of the molecule. Underneath here is adsorbed form, so it's not a condensate, just individual molecules bound. And on the top, you've got a condensed form where you've now got an emergent property of many molecules coming together to form a condensed layer. And this line here, you expect to see a switch-like transition between the adsorbed and the condensed layer. So they decided to look and see whether they could get evidence for this kind of pre-wetting transition by looking at the experiments in more detail. Now, for that purpose, what you want to do is, like many of these phase separation experiments, you want to look at the concentration dependence of the process. How does concentration affect the formation of condensates on DNA? So here's a typical experiment. You can see the KLF4 concentration on the top. You can see the fluorescent microscopy. And you can see as you increase the concentration, you're increasing the number and brightness of condensates. And what they've done here on the lower form is to actually quantify the brightness. And you can see at low concentrations, there's very little, the brightness is quite low, it's quite evenly distributed along the DNA chain. It doesn't get very high above that initial layer. But as you increase the concentration, you now begin to see these condensates popping up. So you're seeing these bright arrays all the way along the DNA chain. And here you can see a particularly nice way that Jose's represented this data, which is here you've got time on the y-axis, just like here, but on the x-axis, what we have is the number of photon counts. And so you can see at a low concentration on the top right here, you can see that you always keep a low concentration of the condensates on the DNA chain. But as you raise the concentration, now you're seeing an accumulation of condensates. So you can see you either have the sort of low concentration or the higher concentration of the condensed state. And here's a sort of nice histogram that Jose's done showing the emergence of the two different populations with time. So this is an individual DNA where he's actually just followed the actual evolution of the intensities along DNA. So what they're seeing then fundamentally is almost a switch-like behavior, you know, either absorbed or condensed on the DNA. And that then suggests that the low intensity corresponds to what I've mentioned, the absorbed state, and the high intensity corresponds more to the condensed state. Right, so here we've got what we call absorbed state. At low concentrations of protein stays pretty similar, but as you raise the concentration, this absorbed state now begins to translate into a more condensed state with many more molecules. And I mentioned these things will have about 1,000 molecules of KLF4 in them, and may have even more. So we can now come back and look at our phase diagram and think that what we're actually seeing when we see formation of KLF4 condensates on DNA is a pre-wetting transition. Is that proteins are normally binding to the DNA and absorbing here. Then, over a certain concentration, you now get a condensed layer driven by the surface. In the absence of the surface, you won't get any condensed layer at this concentration. You have to rather be in this regime to see bulk phase separation. And so this pre-wetting transition was something that we want to look at a bit more and try and understand, could we study the details of what's happening in the absorbed state and what's happening in the condensed state. And for this purpose, we took care of a nice observation that Sina had made that if you attach the MPP to KLF4, this is actually how you purify the proteins and maltose binding protein, KLF4 does not condense, even in bulk, right? It just stays soluble. And actually, for those in the audience who are interested, this is a common trick we actually use to purify these proteins. If you keep the MPP on to the last minute and just cleave it just before you do the experiment, you can keep them nice and soluble during the purification and storage. And so she was able to show that if KLF4 keep the MPP on, there's no condensation at all. But if you chop off the MPP, then you now condense. And so she and Jose decided to take advantage of this to see if they can actually directly watch the switch between the absorbed state and the condensed state on DNA. And this takes advantage of all the tricks of microfluidics. So basically, you take your DNA between two traps, right? So this is how the experiment's set up. You've got your two DNA, which is attached between these two traps, and then held in a microfluidics bath. And then you introduce your MBP KLF4 GFP. Remember, this cannot condense in bulk. And what happens is it binds to the DNA. And you see the low intensity binding, what we call absorption. Then you simply flush in the C3 protease, and that's going to cleave the MBP off in situ. And once you've done that, you can then directly watch the process of condensation under the microscope on the individual piece of DNA. So that's actually a very nice tool to allow us to go measure the types of interactions. And so if you have an MBP KLF4 GFP, so this is one with the MBP on, remember this is the one that will not condense, you measure simple Langmuir binding kinetics. The protein just binds and saturates just as you might expect for a simple absorption. Nothing cooperative here. But if you chop off the MBP, you initially get absorption, because remember the concentration is low, but then you get across this pre-wetting line, now you start to see an increase in the concentration of the protein on the DNA. And that allows you to measure what you call a pre-wetting transition. In this case, a KLF4 of about 100 nanomolar, so about 10-fold less than the bulk phase separation. Remember I said in bulk it's about 1 micromolar, here it's about 100 nanomolar. So we can summarize experimental data then by saying that you have an absorbed state that you can actually measure that binds to DNA in expected ways with Langmuir binding kinetics. You get over a certain concentration and you end up in this condensed state, shown here. And in many ways, it sort of harks back a little bit to some of the old ways that people thought about DNA binding transcriptional factors, which is binding and scanning along the DNA before, when it reached a DNA binding sequence, then forming a cluster that would actually trigger maybe transcription. And so this state may sort of correspond to that sort of initial state where the protein binds and scans along the DNA surface. Now, what I want to say is not, why are we interested in this this aspect of the physics? The biology here might seem a little bit obscure, but actually there are some quite important consequences for considering pre-wetting to actually think about the biology of how these transcriptional factor works, relating very much to the sort of experiments that Phil introduced so nicely at the beginning of the talk. And these are things that people are worried about in the field. One is size control. Another one is that overexpression can change the physical regime. And the other is effects of surface inhomogeneities. So let me go through these and explain how they can help understand aspects of how these transcription complexes can form. First, let's think about size control. If you study phase separation, one of the first things you learn is about coarsening due to Oswald's ripening and fusion, where it's very hard for cells to control the size of condensates because they just grow and grow and grow, and eventually they end up as one drop. And yet we know a condensate on DNA, it actually has a probably defined size. But prewetting actually provides a way of thinking about size control on DNA for the following reason. Let's think about a molecule I've shown in red, close to the polymer. There, it's under surface condensation regime. And so that would correspond to the condenser regime. So you're only going to get formation of the condensation close to the polymer. But now let's think what happens when you get away from the polymer. Now you're no longer under the surface condensation regime. And therefore, the molecule finds itself outside the binodal. And so that means at this stage, you start to get evaporation. In other words, because you only have condensation at the surface, but evaporation away from the surface, that provides you with inbuilt size control of these problems. And in fact, if you go back and look at the formation of KLF for condensates, you see that they get up to a certain size and then they actually stop growing, suggesting indeed that these surface condensation types of mechanisms could be actually a way to limit the size of these surface condensates. Let's now think about overexpression and how that could change the physical regime and what effects that could actually have on regulation of transcription. Because as many people know in this audience, one of the signature aspects of cancer is that many transcriptional factors are overexpressed. This is Isaac Klein kindly made this nice diagram showing that KLF4 and how its expression is in different cancers. But when I come back to think of a pre-wetting pitch, you can see how problematic that could potentially be for regulating the formation condensates on DNA. And let's just go through and think about why that might be. This arrow, the tip, shows you in the absorbed regime, and that's shown in the picture down here. So you just got the Langmuir binding, the molecules evolved to the DNA. Then you increase the concentration, and now you're going to get some surface condensation of the type I've talked about in this talk. And that will give you size control of these condensates. But what happens when you start to overexpress these proteins, as an example? Well, then suddenly you could move into another regime, which is the basic phase separation bulk regime defined by the standard binodal. And that can have sort of two consequences. First, it could lead to formation of drops away from the DNA, but also it could actually lead to spreading along the DNA because you no longer have any size control. And Rick Young told me that, it's quite clear when you overexpress MYC in certain cell lines, you begin to see extension of transcription. And you can imagine that therefore, as you extend down the DNA, you can start to activate genes that you didn't want to activate. And so that means that the concentration will affect very much the physical picture. You might want to look at these particular condensates. And finally, just to talk about DNA sequence and methylation, I've stuck quite clear here about talking about DNA sequences. The reason for that is we've just started to work on that. It's quite tricky to make these 30 kb bits of DNA with specific sequences in them. But from a theoretical point of view, if you go back and look at the experiments I showed you, what I've been showing you is actually individual condensates. But probably physicists in the audience are thinking, but why are they individual condensates? Because actually a classic pre-wetting regime, you'd have initially a low density region, but as you raise the concentration, you'd have a high density, but it would all the way along the polymer. It wouldn't necessarily form these individual condensates. But if DNA polymer, of course, is not homogeneous, inhomogeneous, differently than, say, most polymers studying synthetic systems, of course, driven by DNA sequence elements, but also, as I've shown here, the methylation, right? These things potentially will affect the affinity of the protein. If you look at that from a pre-wetting picture, we've got a homogeneous polymer and a heterogeneous polymer here. You have low density, but if you have, let's say, DNA binding sequence, you have a higher local density here, and then that will actually get you over the pre-wetting transition earlier than if you just have a homogeneous strip, and that might actually give you some form of local condensation driven by the inhomogeneities. And in fact, I don't have time to talk about it today, but Sandeep has done a nice theory on this, and my students that if I give you this QR code, you can actually find a bioarchive paper where he's actually demonstrated very nicely using a Ising model, using DNA states, that you can actually simulate very nicely the pictures that Jose and Sina have seen using their pre-wetting condensation analysis under the optical conditions. So finally, just the thing is that what I've talked to you today is about surface condensation, you know, DNA and microtubules and membranes. And this is going to be a very rich and interesting branch of physics. And I thought I'd specifically talk about it because this is meant to be for students and postdocs and to show you just how much interesting and ununderstood biochemistry is in this problem. I've talked to you about one, which is the interaction of the condensates with DNA, but the way you interact with rigid microtubules will be different than the way they interact with fluid-like membranes. And all of that is likely to be extremely interesting in thinking about how you form these structures. And finally, I'd like to finish by thanking those who did the work, Jose Morin, Sandip Chubai, and Sina Whitman, who are three absolutely outstanding postdocs. And it's a collaboration with a group of Stefan Grill and Frank Ulica. And I thank you so much for your attention.